Right, my dear children. Now, in the earlier chapter, we discussed what are the main products created during the photosynthesis process. And we discussed that glucose is the main product which is getting produced during the process of photosynthesis. Right? And this produced glucose, right, within the leaf is now getting converted into a substance called starch, right? Starch. So it is given in the first sentence here. Now you know that during the photosynthesis process, glucose is the main item or the main product which is getting created. Now in the earlier chapter, we discussed that what are the factors which are essential for the photosynthesis process, and how does a plant is going to absorb these absorb these materials, right? And as well there are three materials, and there is a energy source as well, right? So we discuss how these things are getting absorbed into the plant, right? Then after that we discuss the word equation related to the photosynthesis process, and we discussed that the glucose is the main component which is getting produced during the photosynthesis. So after that, now we are discussing what are the other contents related with the lesson part. So here it is given that glucose, right? So you know that glucose is the main product which is getting created within the plant, okay, during the process of photosynthesis. So glucose which is getting produced here, right, is not going to be it's not going to be transported all around the body parts of the rest of the body parts of the plant. So the glucose which is getting produced within the leaf is getting converted into a substance called starch. Starch is a type of a carbohydrate, right? Starch is a type of a carbohydrate. Actually, starch is made up with collection of several glucose molecules. With the collection of several glucose molecules each other, starch is getting produced, right? Several glucose molecules joins together to form starch. So after the photosynthesis process, you know that glucose is getting produced within the leaves, cells of the leaves. So with the production of glucose molecules, right, with the production of glucose molecules, these glucose molecules are getting together, or they combine together to form a special kind of a structure, special kind of a compound which is referred as starch. Okay, each and every carbohydrate is made up with glucose. Glucose is the building unit of carbohydrates. So starch is also a type of a carbohydrate. So specialty here is that this carbohydrate or the starch is made up with collection of several glucose molecules. So my dear children, remember the glucose which is produced under the photosynthesis process in the leaves is converted into starch. Now it is getting converted into starch. It's not going to happen like sudden, but however, upon the production of glucose, time by time, all the glucose, right, all the glucose is getting collected together and it's getting converted into starch. That's how this is going to work, right? So temporarily, starch is getting produced within the plant leaf if photosynthesis is going to occur, right? Temporarily, it's getting stored as starch within the plant leaf itself. Okay, so now you can get an idea that how does animals, like herbivore animals, get their energy for their day-to-day activities. They also receive by eating plant items, no? So you know that plant leaves containing starch. Starch is a good carbohydrate. So the main meal received by herbivore organism is plant leaves. So plant leaves, as plant leaves contain carbohydrates, carbohydrate is the main food item or the main food dish received by the herbivore organisms. You know that even us, even animals, we also receive carbohydrate as the main source of nutrition in our meals. If you eat rice, rice contain carbohydrate. Bread, main ingredient is carbohydrates. Hoppers, string hoppers. All of these foods mainly contain carbohydrates. So carbohydrate is the main food item, main nutritional food item contained within each and every food item that we consume, right? Main food item. So there are some others as well. If you eat meat, most of the times you'll be receiving proteins, and fish also proteins. But however, I'm talking about normal day-to-day. Now, no, those things are special things. No, most of the food that we eat in our day-to-day life, like bread, rice, then after that yams, all of these things mainly contains carbohydrates. So in plant leaves also these carbohydrates are present. In plant leaves also these carbohydrates are present. The plant leaf is having a carbohydrate called a starch. Okay, starch. So starch is getting produced within the plant leaf by collection of several glucose molecules, by collection of several glucose particles, which is produced during the process of photosynthesis. Okay, which is produced during the process of photosynthesis. So my dear children, upon happening photosynthesis within the plant leaf, the glucose which is getting produced is getting stored temporarily within the leaf in the form of starch. Okay, in the form of starch. Starch is a good carbohydrate. Okay, starch is a good carbohydrate. Now in grade 7 also, I think you have got a sound knowledge about the starch and how to identify starch. We use iodine solution to identify starch. So we'll be discussing about those things in this lesson part as well, right? So however, remember that starch is the main food item contained within the leaves. Okay, how does leaves is going to receive this starch? Because of the photosynthesis process, right? During the photosynthesis, glucose is getting produced. These glucose then collect each other. Several glucose molecules collect each other in order to produce starch. So starch can be identified temporarily in plant leaves if photosynthesis is going to occur. Okay, right. Later, this starch is converted to sucrose and transported to the necessary places of the plant, growing parts and storage organs. Now, here actually we are discussing what is going to happen with that glucose which is getting produced within the plant leaf during the photosynthesis process. So first and foremost, what's going to happen here is, during the photosynthesis process, you know that glucose is going to be produced. That produced glucose is getting collected each other to form a carbohydrate called the starch. Then after that, there is a certain level, or there is a certain amount which the starch can be kept within a plant, right? Plant means actually a plant leaf, right? So within a plant leaf, there is a certain limit. So after that limit, what's going to happen? The produced starch within the plant leaf is getting converted into a special solution, right? Special solution called sucrose. It is getting converted into sucrose solution. Sucrose means simply sugar, right? Chemical name of sugar is sucrose. So my dear children, glucose is getting converted to starch, which is a carbohydrate. So that carbohydrate is now getting converted into a sugar solution or else sucrose. Okay, so that's why I told you that glucose, sugars, carbohydrate, all of these things are made up with ultimately glucose, right? So that's why I told you that in the earlier chapter, you could remember, I think, carbohydrate have different properties rather than sugars. Sugars have different properties rather than glucose. But all of these things are made up with glucose. Upon change in the number of glucose particles, these properties change, right? Upon change in the glucose particles, these properties are going to change. That's how it's going to work. Okay. So my dear children, all of the foodstuffs that we are going to consume mainly contain carbohydrates. Each and every carbohydrate is made up with glucose. Even the sugars are made up with glucose. So what happened here is the glucose which is getting produced is getting converted into starch within the leaf. Then after that, it's again getting converted into a sugar solution or else to a sucrose solution. That's the chemical name. Then finally, this sucrose solution is transported to the necessary places of the plant, right? Necessary places of the plants. So starch is a carbohydrate, so therefore it's difficult to transport. So the transportation process is going to occur mainly by converting the starch into sucrose. Sucrose is readily dissolving in water. You know that sucrose means sugar, right? Sucrose means sugar. So it's getting readily dissolved in water. So there is no problem in transporting sucrose solution throughout the plant, as it is getting well dissolved within water, right? So my dear children, the starch is getting converted to now sugar solution or else to a sucrose solution. Then after that, this sucrose solution is getting transported to the necessary places all around the plant for each and every cell, right? All around the plant parts, these sucrose are going to be transported. The excess sucrose solutions, excess ones, after utilizing these things necessary, right, in the necessary level, excess ones finally getting converted into or getting transported into storage organs. Storage organs. Mainly they are being transferred into growing parts. Then after utilizing for the growth and any other resources or any other processes, the excess amount of sucrose then finally getting transported into storage organs. Storage organs like yams. In sometimes roots, underground roots, sometimes aerial stems like sugarcane, underground stems, like underground roots, beetroot, carrot. Okay, you know that beetroot we can consume, carrot we can consume. Those things are roots. Okay, those things are roots, storage roots especially. Here the sucrose solution is getting transported to those roots and stored as carbohydrates once again. Okay, once again stored as carbohydrates and getting stored within the roots. Sometimes those may transfer into different other places like underground stems. Okay, in underground stems like ginger, right? So these things are being transported to the necessary underground stem and it is being stored as starch. Starch or any other carbohydrate. Okay, sometimes onto the aerial roots like sugarcane. So within sugarcane, you know that the stem of the plant can be consumed, or we can consume that thing. I mean, like, we can eat it, right? And we can use the sugar within it. So sometimes there are yams, fruits especially, right? Yams and fruits. These are the storage agents. So these are on excess carbohydrates or excess sugar solution or excess sucrose, which is getting transported in after to the necessary parts of the plant, will be stored as starch once again, or starch or any other carbohydrate once again within the storage agents in the plant. Okay, those storing agents are the ones that we are going to consume, especially fruits, yams, aerial roots, right? Aerial stems, then after that underground stems. So those are the ones which are referred as storage agents. So within these, the food which is getting produced by the photosynthesis process is going to store its necessary amount of food. Okay, excess food. So plants are giving us the food that we want. Okay, they are not going to expel the excess food. They are providing food for the existence of the organisms as well. So that's why it is being referred that plants are the main one which is providing, main living component which is providing the security and which is going to contribute for the existence of life in the earth. They are going to produce food by themselves, and also they are going to produce, the produced food is being given to the animals as well, right? So this is how simply the transportation process is going to work, starting from glucose. Glucose is getting converted to starch, which is a good carbohydrate, temporarily getting stored within the leaves as starch. Then after that, it is once again converted into a sugar solution or else to a sucrose solution, then transported all around the plant. Growing plants are growing. Then after that, excess sucrose which is getting stored within the plant is being transported to the necessary storage agents. That's how the food is getting stored within a plant, right? So it can be any storage organ, like a root, aerial stem, underground stem, fruit, any kind. So we consume those parts in a plant in order to get food that we want. Okay, so this is how the photosynthesis process is going to occur, and the transportation of food which is going to happen within the plant, and also distribution of food to the environment, the other organisms in the environment is going to occur, right? Okay then, so next point is given. Let us do an activity to test whether photosynthesis has taken place in leaves. And now we are going to conduct an activity to show that photosynthesis has occurred within the plant leaf. Okay, now we are going to show that a photosynthesis process has occurred within a plant leaf. So my dear children, in order to show that photosynthesis has occurred within a plant leaf, what we are going to do is we are going to test the plant leaf for starch. We are going to test it for starch. Okay, so if a starch is there, definitely photosynthesis has occurred. Why is that? Because there is no possible way to form starch without glucose. There is no possible way to create a starch without glucose. The only living process which is able to produce a starch is by glucose only, and glucose is getting produced by the process of photosynthesis. So my dear children, here we are not going to directly show that glucose is getting produced within the plant leaf, but however, we are saying that, or we are showing that starch is getting produced. If there is starch, definitely there is glucose, because there is no possible way to create starch without glucose. So glucose is produced upon photosynthesis, we already know that. So thereby here we are showing that starch is present within a plant leaf. By that way we can come into a conclusion that the starch is there. If starch is there, then we can say finally glucose should have to be there. If glucose is there, definitely photosynthesis has occurred. So here once again remember we are showing that a starch is present within the plant leaf in order to show that photosynthesis has occurred, because each and every glucose molecule which is getting produced within the plant leaf is temporarily stored as not glucose but as a starch form. So if the leaves contain starch, then we can come to a conclusion that photosynthesis has taken place in the leaves. So if there is starch, then we can say that there is photosynthesis. So it's really simple. What's going to happen? Photosynthesis is going to produce glucose. Then collection of several glucose is going to produce starch. So this is the main component which is getting stored within the plant leaf upon photosynthesis, right? So we are testing whether the starch is there or not. If there is starch, then definitely there should be glucose. If there is glucose, yes, the photosynthesis has occurred. Okay. If there is no starch, then no glucose, then no photosynthesis. Okay. So we are testing whether starch is there or not. So by doing this activity, we'll be preparing a plant leaf for the test of starch. So let's see how to prepare a plant leaf for the starch test. Right. So my dear children, you are given with the activity investigating whether leaves contain starch. Remember, we are checking whether there is starch, not glucose. We can't identify glucose within plant leaves because glucose is getting converted to starch. So we are checking whether starch is present within the leaves or not. Okay. So you'll be needing these materials. Right. So you'll be needing these materials when doing this experiment. Right. Now let's see what are the materials that we need. So you'll be needing water, water beaker, right? A water beaker. Then after that, I'll name the main ones first. We'll be needing a tripod, then a Bunsen burner, or spirit lamp. In order to heat. Then after that you will be needing boiling tube. Definitely you will be needing iodine solution. This is, my dear children, this is alcohol solution, right? Alcohol solution, or just name it as alcohol. You'll be needing some alcohol as well. Okay, alcohol. Then definitely you'll be needing a green plant leaf. We'll be needing green plant leaf. why do I need to mention it as a green plant leaf? Because only in green plant leaves chlorophyll is present. There is only chlorophyll, then the photosynthesis is going to happen, right? So green plant leaf is really important. Don't take a plant leaf which has any other different colors in here. Okay, there are other plant leaves which has different mosaic colors, right? We can't use those things. Like croton, we can't use that kind of a plant leaf. However, it should be a green color one, right? These are the main things that we need to, right, find when you are preparing this leaf to plant test the starch test. Okay, so my dear children, these are the main parts or the main materials that we need. So there are two other materials. Okay, so now here in this process we are heating a boiling tube now. So we'll needing this boiling tube in order to heat the plant leaf. Okay, I'll mention the use of alcohol when I'm going to do the method, or when I'm conducting the activity. When I'm explaining about the method, I'll explain why do we need alcohol. Okay, so now we need to boil this boiling tube, right? So you know that in order to take that boiling tube out, or in order to hold that thing, we'll be definitely needing a holding instrument. So this is our forceps. Forceps. Okay, we'll be needing forceps. Then after that, in order to get a clear observation, in order to get a good clear observation, we'll be needing a porcelain tile. If it is good, then that one is a white one. If it is a white one, then it is very good. Okay, but however, don't choose a dark color one. Go for a light color porcelain tile. So white tile is good. White color tile is good. White or light color tile. Okay, so actually this thing is, if you can't find out this thing, you can just use a piece of paper, A4 sheet, right? If you can't find out a white color tile, then we can directly take out a simple piece of paper and place the leaf on it. Then we can do the test that we want to get clearer observations, right? So light color tile. Light color tile. So light color means actually white would be great, right? If you don't have a white color one, you can use any kind of a light surface. Okay, so light color tile. So these are the materials that you need. Okay, so you'll be needing a water beaker, then white or light color tile, tripod, Bunsen burner or else a spirit lamp, and green plant leaf, some alcohol, boiling tube, iodine solution, and pair of forceps to handle our boiling tube. Right, now let's see how to conduct the activity. So you are given with two different figures. First figure, boiling in water. Second figure, B, boiling in alcohol in a water bath. Right, so these are the two main methods. Now I am going to explain, my dear children, how to prepare this plant leaf for the test of starch. Okay, now listen very carefully and get the knowledge on preparing a plant leaf for the starch test, right? In your O levels also there is a lesson called photosynthesis at grade level. So in that lesson part also we'll be discussing this entire experiment in detail. So this is the foundation for you guys. So listen very carefully and get the idea on doing the experiment. There are several steps. Okay, so I'm going to explain step by step in how to prepare this plant leaf into the test of starch. Okay, now let's see what is the first and foremost step. It's really simple. You just take the beaker. This is the beaker, right? You just take the beaker in the first step and dip the green leaf inside the water beaker, and you have to boil it very well. You have to boil it very well. In a plant leaf, right? In a plant leaf, you know that there are cells. In a plant cell, my dear children, there is a special structure called cell walls. Cell wall. If this is a plant leaf, then this is the one which is called as the cell wall. Cell wall is going to give out a special protection for the plant leaf cells. Not in plant leaves, but also in each and every cell. Most of the cells we can observe in a plant, most of the cells we can observe this cell wall thing. But in an animal cell, you can't observe a cell wall. The animal cell is like this. There is no cell wall, right? There is no cell wall in animal cells. But in a plant cell, for each and every plant cell, there is a cell wall, right? There is a cell wall. Usually plant cells are like more having a polygonal shape like this. It's having a polygonal shape like this, right? So there are different structures within the plant cell also, like this way. In animal cells also there are different structures, right? Okay, so you know that these things are the ones which are referred as organelles. In a plant cell, there is a protective organ or organelle which is called as the cell wall. So my dear children, it's very hard to get an observation. You know that starch is produced within the, now let's imagine that this is the, right? So this is the one which is called as chloroplast. You know that within the chloroplast, right? You know that within the chloroplast, starch is getting produced or glucose is getting produced. So my dear children, you have to break this cell wall like this. You have to break it down. Then you have to expose these things. Then you have to expose those things, right? Okay, so you have to expose those things like this without breaking the cell walls. Without breaking the cell walls, you can't expose the contents within a plant cell. Animal cells are quite easy because they do not have a cell wall. So I'm going to erase this thing as it is not necessary. Plant cell is somewhat way more different than the animal cells. It contains a special protective layer called a cell wall. So first and foremost, we have to break this cell wall and we have to expose the things inside the cells. By that way only we can test or we can identify these chloroplasts, and by that way only we can add whatever the stuff that we are going to put into directly chloroplast. By that way we can check whether there is starch or not. If you just add iodine into these, then it will be not added into these structures, especially for chloroplast. Now we need to add iodine into the chloroplast. That's the place where glucose is getting introduced. That's the place where starch is there. So in order to expose chloroplast, we have to remove the cell wall around it. So we have to remove it. Like this, we have to remove the cell wall. And we have to break it down like this. Then you know that now. And now we have exposed chloroplast. Now we can add whatever the component that we need to test. Okay, whatever the component that we are going to add for the test of starch. Now we can add on to the chloroplast. But there is another difficulty. What is that? You know that chloroplast contain chlorophyll. Right? Chloroplast contain chlorophyll. Chlorophyll is in dark green color or green color. As it is having a green color, it's very difficult to get an observation. We can't clearly observe. It's very difficult to get an observation. So then we have to remove the chlorophyll within the chloroplast as well. Right? So we have to remove the chlorophyll also. After removing the chlorophyll, then the leaf is going to have some kind of a faded light color. Right? Light faded color could be observed within the plant leaf. If you are going to remove, chlorophyll, now the first and foremost thing is to break down all the cell walls within the plant cells and exposing these organelles within each and every cell by breaking down the plant cell walls within the cells. Then after that, we have to remove chlorophyll. We have to remove chlorophyll to get a good observation. Alcohol is a wonderful solution for chlorophyll. Chlorophyll is not getting dissolved like a substance or like water-like substances. Okay, chlorophyll is not getting dissolved like water or in a watery medium. Water-like substances cannot dissolve chlorophyll. You know that there are certain substances in our environment which can be dissolved within water and which cannot be dissolved within water. For an example, if you take grease, oils, those things are not getting dissolved within water. It's mainly because grease and oil and all the other things are made up with carbon-based materials. In simple, these things are made up with mainly from plant and animal materials. Each and every oil type is made up with carbon-based materials or plant and animal materials. So those things are not going to dissolve in water because water do not contain any carbon materials, right? So therefore water is referred as an inorganic material. If there are carbon or carbon as the main, if we can identify carbon as the main constituent, then those things are referred as organic substances. If there are no carbon, then we refer those things as inorganic substances. Water, you know, there is one oxygen and two hydrogen atoms or particles. So therefore water do not contain carbon. But however, chlorophyll contain carbon. Chlorophyll is a type of a substance made up with mainly from carbon. So we can't dissolve chlorophyll in water. As they are unlike substances. They are unlike substances. So we have to get a like substance in order to dissolve chlorophyll. If you take Jack glue is also the same. Jack glue is not going to dissolve in water, but however, Jack glue is going to readily dissolve within coconut oil or kerosene oil because all of these oils are made up with carbon-based substances. Fossil fuels, now we make kerosene oil from fossil fuels. Fossil fuel is made up with carbon-based substances. Plant and animal materials, no. Coconut oil also the same. Coconut oil is being obtained from coconuts, a plant material, and they contain carbon. So carbon-based substances are readily getting dissolved within the carbon-based substances. But water is not a carbon-based substance. So therefore you can't dissolve chlorophyll. So my dear children, in order to dissolve chlorophyll, we have to get a carbon-based substance which is alcohol. You know that alcohol is being received by the fermentation of sugars or carbohydrates. So it is a carbon-based material. So in order to dissolve chlorophyll, we use alcohol. But it's not going to just dissolve. You can't just put alcohol into this thing and to dissolve chlorophyll. We have to place this leaf on the solution of alcohol, then we need to boil it. So second figure includes that thing. Okay. In the first figure, we are going to break down the things to break down the things which are referred as cell walls and to expose the chloroplast within the cell. Then in the second step, we are dissolving chlorophyll in order to get a good observation. Right? The other thing is that boiling in water and boiling in alcohol doesn't affect the starch contained within the chlorophyll. Okay. So after these two processes, right? After these two processes, my dear children, our leaf is going to be prepared for the starch test. Now, chlorophyll is being removed, cell wall is being removed, and the chloroplast is now exposed to the outer environment. Now we are going to add our indicator. Our indicator is iodine solution. Now we are going to add the iodine solution onto this plant leaf. By that way, if there is starch, a color change is going to occur. By checking that color change, we can say whether the photosynthesis has occurred within the plant or not. Right? Actually, we are going to say that the presence of starch, you know that if the presence of starch can be proved, then we can say that the photosynthesis has occurred. In the earlier chapter, we discussed that thing. So, my dear children, this is the way of doing, or this is the way of preparing our plant leaf for the starch test. So, I'm going to erase this part, then we'll write it down step by step. What's going to happen? So, first of all, what do you have to do? Arrange the setup. Arrange the setup. Arrange the setup according to figure A. According to figure A, you have to arrange the setup and boil the leaf very well, within water. Right? So this is the first step. You have to arrange the setup according to first figure. Now this is the Bunsen burner. Okay. So first of all, you have to turn on the Bunsen burner, and according to the figure A, you have to turn on the Bunsen burner and you have to heat it very well, right? So arrange the setup according to figure A and boil the leaf very well within water. We have to boil it very well within the water. First step. Okay. Right. Then after that, why do we need to boil it in water? In order to break the cell walls and expose the things or the organelles to the outer environment. Right. Next step. Then we have to remove the leaf and place it on that boiling tube. We have to remove the leaf and place it on that boiling tube. Then we have to put some alcohol solution into it. Okay, then we have to put some alcohol into it. Then don't directly heat alcohol. We can't directly heat alcohol, my dear children. It's really important to remember that thing, because alcohol is a highly combustible substance. If you're going to directly heat alcohol by applying, we are going to remove that water, and now we are going to heat it, like heat the alcohol solution by keeping like this way. So if you are going to do so, definitely this alcohol solution or alcohol container will catch on fire and you might get burned. Sometimes it may explode, right? So don't do that thing. Remember, this is really important. We have to dip the alcohol solution within a water beaker. That is for our safety. That's a safety precaution, because alcohol is a highly combustible substance. Right? Alcohol is a highly combustible substance. So that's why we need to dip that thing within the water beaker when you are heating or when you are boiling the plant leaf within alcohol. Remember that thing is very important. We can't directly heat alcohol as it is highly combustible. So in the next step, what you have to do, you have to take the leaf out and place it in the boiling tube and add some alcohol solution. Okay, so we'll write it down. Take the leaf out, dip it in boiling tube, with alcohol. Take the leaf out, dip it in boiling tube with alcohol and place the boiling tube again in water beaker and boil the leaf in alcohol very well as per to figure B. Right. So this is our second step, my dear children. What is the second step? Take the leaf out. Now we have to take the leaf out from the beaker. Dip it in boiling tube. We need to dip it in boiling tube. Right. Then with alcohol, and it should contain alcohol, means that you have to add some alcohol into the boiling tube. Okay. So take the leaf out, dip it in boiling tube with alcohol and place the boiling tube again in water and boil the leaf in alcohol very well. Right. I'll mention this thing as water beaker. Right. I'll mention this thing as water beaker. Right. So take the leaf out, dip it in boiling tube with alcohol and place the boiling tube, right? Boiling tube Place the boiling tube in water beaker and boil the leaf in alcohol very well, as per to figure B. Now this is the figure B, this is the figure A, right? I hope that the two steps are very clear. Once again, let's see. Arrange the setup according to figure A and boil the leaf very well within water. Within water, first of all, you have to boil the leaf. Second step, take the leaf out. Dip it in boiling tube. Take the leaf out, dip it in the boiling tube with alcohol. So it contains alcohol now. And place the boiling tube in water beaker. Now we have to remember this step. It's really important. We have to place the boiling tube within the water beaker. Otherwise it will explode and you might get burns. Okay. So remember that thing. So place the boiling tube in water beaker and boil the leaf in alcohol very well as per to figure B. So according to figure B, you have to heat the system very well. Okay, right? First one, it helps to break the cell walls. Second one, dip it in alcohol. It helps to dissolve chlorophyll. Now our two main steps are over. Third step. I'll take the slide somewhat up. Right. Third step. Take the leaf out. Wash it with water. So we have to wash it with water as there is alcohol, right, on the surface of the leaf. We have to first of all wash it with water. Okay. So take the leaf out, wash it with water, then add few drops, of iodine solution. Add few drops of iodine solution. Add few drops of iodine solution. Observe the changes. Observe the changes, right? So this is the final step, right? So there are 3 steps. First one, you have to boil it in normal water, right? You have to boil it in normal water very well. Second step, you have to take the leaf out, place it in the boiling tube, put some alcohol, place the boiling tube in the beaker, then boil. Don't directly heat alcohol because it is highly combustible and the boiling tube may explode. Okay, right? Then after, you have to take the leaf out, wash it well with normal water. Then we're going to add few drops of iodine solution, right? So these are the 3 main steps. Finally, adding iodine solution, we need to observe the color change, my dear children. So this is how to prepare a leaf for the starch test. If you are going to follow with these steps quite clearly with the given instructions, finally you will obtain not a green color leaf, but a leaf that has a faded light color. And if you have correctly conducted the experiment, definitely this boiling tube over here this boiling tube over here, this one would have a good greenish color. Definitely this one would have a good greenish color. Okay, when I am doing this activity, I am going to show you that thing, that the alcohol in boiling tube, it's going to turn into greenish color because our aim is to dissolve chlorophyll. So chlorophyll is green in greenish color. So after dissolving chlorophyll within alcohol, definitely this color of alcohol solution should turn to green color. As there are no chlorophyll particles within the plant leaf, after removing the plant leaf from the boiling tube, it should contain a faded light color, right? So my dear children, these are the 3 steps, and this is the way of conducting our experiment, or this is the method of conducting or making a plant leaf into the experiment of iodine test or else starch test. So this is how to prepare the plant leaf for the iodine test or for the starch test, right? So now my dear children, it's time to really do this activity at our laboratory. Now let's head on to the lab and we'll conduct our experiment. We'll prepare the plant leaf for the starch test and let's see whether we can get the necessary observations or not. So my dear children, in this activity, we'll be investigating the presence of starch in a plant leaf. So to do this activity, I have several instruments with me. So first of all, I have prepared the instrument in here by using a tripod and a mesh and our water beaker in order to heat the plant leaf. So I have a crucible I have a forceps with me, then a small bit of a forceps in order to handle these leaves, right? So I have 2 green leaves with me here, then a boiling tube, after that some alcohol, right? Ethyl alcohol, ethanol. So first of all, what I'm going to do is I'm going to take these 2 leaves and insert it to the water like this, right? Now what you have to do, you have to boil it very well. So let's slide it up. Okay, now we have to boil it very well. Right, so let's keep the system for a while until the 2 leaves are going to boil. Okay, so we have to boil it very well in order to remove the cell walls and to be open and to open the chlorophyll. So after that, we are going to put the 2 leaves inside the boiling tube. Then after that, I'm going to add some alcohol. Then we have to reheat in order to remove the chlorophyll. So with the chlorophyll, you know that we can't clearly get an observation. So therefore, we have to remove the chlorophyll in the system of the leaves. So let's keep these leaves for a while so that they would boil up and to expose the cell walls to remove the chlorophyll. So as you can see here, now the water is starting to boil. So we have to insert the leaf within the system so that the leaf also boils very well. Right, so now you can see the water has started to boil, right? So you can see the bubbles over there, and now we have to boil the leaves very well within the water, right? So you have to insert the leaves within the water and we have to boil it very well for a few minutes. right? So once again we'll keep the system in order to boil for a few minutes. Then after that we'll take it out and let's boil it inside the alcohol. So we'll keep it for some time in order to boil it very well, right? So now as you can see here, now the two leaves are well boiled. So now I'm going to take it off and place it within this boiling tube. So you can see that the color of the leaf has got changed right now, right? The color has got somewhat changed. See, you can see that it has like brownish color nature. Okay, so now I'm going to place this larger leaf into my boiling tube like this. We have to keep this turned on. You know that we have to boil it again by using alcohol. So let's insert the second one. Right. Now what you have to do, you have to add some alcohol into the boiling tube. So you have alcohol with me. I have alcohol with me right now. So I'm going to insert some alcohol. Alcohol is highly flammable. So it's wise to keep away from the fire, and we'll insert. Right. So I'm going to wipe it down. So my dear children, now don't boil alcohol directly under a flame. It's because that you know that alcohol is highly flammable. So when you are inserting this thing, make sure you insert this thing within water like this. Right, now we need to allow it to boil somewhat so that the chlorophyll will mix very well with the alcohol. So we'll allow it to boil somewhat. As you can see here now, see, the solution is turned into greenish color. It's because that chlorophyll is getting mixed very well within the alcohol solution. So chlorophyll, in order to remove chlorophyll, we need to use alcohol. So that's why we need to boil it inside alcohol. But however, you can't directly boil alcohol is because that alcohol is highly flammable. So if you directly heat it, there might be a possibility that it would, you know, like explode and you might get hurt. So you have to be very careful when you are heating alcohol. Right, so you can see now the solution has got turned into a greenish solution. So the smaller paper now has turned into like white color. So let's keep the system for a while, and after that we'll take the two leaves out. Right, my dear children, now I'm going to stop the boiling process. So first of all, I'll put off the flame, right? So let's take it out and let's put off it. So before that, we have to take the boiling tube out. So I'm going to remove this system away. Right, so now what I'm going to do is I'm going to remove the two leaves here, right? So let's see what has happened over here. So I'm going to use that forceps thing. Let's take somewhat up. So as you can see here, now the leaf has got turned into a white color. So we'll keep it on the watch glass. So as for to the larger big leaf, it doesn't change that much into a white color. So therefore we'll use the smaller leaf, this one, in checking for the presence of starch, as it turned into completely white color. So we'll leave it here, and you can see that so solution has got turned into a green color. So that it's because of dissolving chlorophyll within the alcohol solution, right? So I'm going to remove it now. Let's keep it in here. Now before we are going to add starch into this one, we need to wash the leaf very well, right? So I have a water beaker with me. So you have to place the leaf and we have to wash it very well, right? So in order to dissolve all the alcohol, so dissolve alcohol, I'm going to wash it very well. Right. Okay. Now let's take the leaf out. Now this is my plant leaf which I have prepared for the starch test, my dear children. So what are the procedures? Once again, we boiled using the normal water. Then after that, we took it out and we placed it in a boiling tube, and we filled the boiling tube with alcohol. Then we placed it inside the water beaker. Then once again, we boiled it. Okay. So after that, now I'm going to test this system for the presence of starch. Right. So my dear children, I have cleared my desk so that you can observe the leaf very well. So now this is my leaf which I have prepared for the starch test. Okay. Now it has a like white, like a faded color, not exactly white, but however you can observe that the color has got faded somewhat. Now I'm going to place it over the white porcelain tile so that you can observe the color change very well. So I'm going to remove the watch glass. Right, now what I'm going to do is I'm going to add some drops of iodine solution into the leaves. Right, now let's observe what's going to happen. So I have an iodine solution with me and I'm going to use a dropper also, right? So see, so this is the normal color of iodine. It has a brownish yellow color, right? Brownish yellow color. So let's add some iodine drops into the leaf. So actually you have to spread it here and there, right? As you can see here, now the color is getting changed, right? So you can clearly observe the color is changing. Now the color has got changed into purple blue color. You can see, you can clearly observe that the color of the iodine has got turned into a purple or blackish blue color. See, it has turned into a blackish blue color. So what I'm going to do is I'm going to take once again some drops of iodine to show the color difference for you guys. So check the difference in colors very well. See, this is the original color, the brownish yellow, and in here you can observe the purple blue color, right? it has a dark blue color nature, right? So with this test and from this observation, my dear children, we can finally conclude that the leaves is producing starch when there is photosynthesis. So if the photosynthesis has occurred within the plant leaf, then by only we can identify the presence of starch. So we can say that within this leaf, the photosynthesis has got occurred. That's why the color has got changed into purple blue color as per to the activity. So finally we can conclude that photosynthesis has occurred within the leaf and a starch has been produced. It's because that the solution of iodine or the iodine solution is getting converted into purple blue color or dark blue color with the presence of starch. Right, then let's head on to the classroom once again and we'll study the further things related with the lesson. So my dear children, we conducted the activity and you could see that when I was taking that leaf out from the boiling tube, it's having a faded light color apart from the greenish color, and the boiling tube had a greenish color because it's containing chlorophyll right now. So when I'm going to add that solution, iodine solution, into the plant leaf, you could clearly observe that there was a blackish blue color after a few seconds. A blackish blue color. We refer this thing as also a purple blue color. Blackish blue or purple blue. Both the things are correct. Huh? So my dear children, here we could clearly observe that starch has been produced. Iodine is giving out that blackish blue color with only starch. If there is only starch, then iodine color is changing into blackish blue from yellowish brown. Normally it has a brownish yellow color. Okay, so when iodine is added to starch, that brownish yellow color is changing into purple blue or blackish blue, right? It is changing into blackish blue. Usually I have shown you that usually iodine is in brownish yellow color. However, it is turned into blackish blue with the presence of starch only. With the presence of starch only. If there is no starch, then you can't observe that color change. It's not going to occur with any other substance. Only with starch that color is given. Right? So my dear children, here you are given with the figure as well. What do you have to do? You have to take that dropper thing and place the dropper on the iodine solution. Then this is the original color of iodine solution. You can see it very clearly. It is having a brownish yellow color. Then we have to add that we have to add few drops of iodine solution onto the surface of the leaf that we have prepared for the starch test. Then we have to observe the color change. Okay, then we have to observe the color change. So color change is quite simple. Brownish yellow color is changing into blackish or purple blue color, right? This is our observation. So we can write down our observation. So observation is quite simple, my dear children. Observation is quite simple. Here what's going to happen? Brownish yellow iodine solution turns to blackish blue purple blue. Right? So brownish yellow iodine solution turns to blackish blue. Okay, so this is our observation. Okay, right? Brownish yellow iodine solution turns to blackish blue or purple blue. So what is the conclusion that we can take from this activity? So conclusion is very simple, my dear children. Blackish blue color is given, blackish blue color is given by iodine, right? Blackish blue color is given by iodine with starch only. With starch only. So blackish blue color is given by iodine, right? Blackish blue color is given by iodine with the starch only. Only with the starch, this blackish blue color is given. Therefore, leaf contains starch. Therefore, photosynthesis right? Therefore, photosynthesis has occurred within the plant leaf. This is our final conclusion. Specialty here is that once again I'll mention blackish blue color. This blackish blue color is given by iodine with a starch only. Only with the starch, blackish blue color is given. So therefore, leaf contains starch. If blackish blue color is given by iodine, then we can say that leaf contains starch. If leaf is going to contain starch, then we can say photosynthesis has occurred. How come? Because glucose is the one which is responsible in creation of a starch. Glucose is created because of photosynthesis. So my dear children, finally we can conclude from this activity that the leaf has undergone with the process photosynthesis because the leaf contains starch. Starch is created only due to having glucose, right? So this is how we can prove that photosynthesis has occurred within a plant leaf. Okay, this is the way of proving that the photosynthesis has occurred within a plant leaf, my dear children. So once again, blackish blue color is given by iodine with a starch only. Therefore, leaf contains starch. Therefore, photosynthesis has occurred within the plant leaf, right? So this is our final conclusion from the activity. This conclusion can be taken according to the above observation. because brownish yellow iodine solution turns to blackish blue, right? So this is our final conclusion from the activity, right? So here leaves turn into dark blue color. Dark blue means blackish blue, I told you that, or purple blue. So leaves turn into dark blue color when iodine is added. Iodine is an indicator, so it is an indicator that turns to dark blue with the presence of starch. So with the presence of starch, it is getting turned into dark blue color. So you can observe the color in here. I have given with the figures, right? So this is positive. If the starch is positive within a certain type of a material or else a solution, then this is the color which is given by the iodine solution, right? And this is the original color of the iodine solution, normal brownish yellow color. So this is negative, right? Negative means there is no starch. So when you add this kind of a solution to this kind of solution, this kind of a substance, it is getting turned into blackish blue color or dark blue color, but there should be starch, right? There should be starch. So this is the color change which is going to occur, my dear children, right? So when starch is absent, you can observe the normal brownish yellow color like this. When starch is present, it is turning into a blackish blue color like this, right? Dark blue color like this. So this experiment demonstrates that when necessary conditions are supplied, the leaves perform photosynthesis and produce starch. So when necessary components are given to the plant, right? Plant leaf is going to do the photosynthesis process and produce glucose, thereby starch. Collection of several glucose together is going to form starch. However, starch is going to be produced if there is only photosynthesis. So my dear children, I have provided you several figures so that you can get a good knowledge about the color change as well. So here, this is the color change that is going to occur. So we can test leaves for the presence of starch using iodine solution. So this is what going to happen with the iodine solution. If starch is not there, then it is going to show this kind of brownish yellow color. If it is present, if the starch is present, then it is going to provide a blackish blue color. So a real figure is given in the other side for the starch test. Okay, this is the positive color and that is the negative color, right? Negative color means normal color of iodine solution, right? So this is the way of preparing a plant leaf for the starch test, my dear children, and how to test that plant leaf for the starch. The indicator that we use is iodine. Upon having starch, the color of iodine is getting changed into blackish blue color from brownish yellow, blackish blue or dark blue, right? So I hope that you got a good knowledge about preparing a leaf for starch test and to show that how the photosynthesis is going to occur within a plant leaf. So here we have proven that photosynthesis has occurred within the plant leaf that we took. Okay, right then. So we'll be discussing further things related to our photosynthesis lesson part within our next chapter. So I hope that you will remain with me until the end of the lesson. So in the next chapter, we'll be mainly focusing about the oxygen gas which is getting produced during the process of photosynthesis. Then I'll hope to meet you with our next chapter to discuss further parts with the lesson.